तो ऐसा ना टाइम पे मत लगे आपके पास समझे कर लेते कर कॉपी उधर उसमें कर
this audience. And to honor the discovery of Raman effect by the Nobel laureate, physicist Sir C.V. Raman on February 28, 1928. And that also earned him the Nobel Prize in Physics in 1930 and marked a major milestone in Indian scientific achievement. Now, on this very special occasion, we have with us Professor Uma Ramakrishnan from NCBS Bangalore. And I request our director, Sir M. Dr. Prakash Kumbare, to please escort the dignitary to the dais. Coming back to the day, and as you know that the official theme for 2026 is Women in Science, Catalyzing Viksit Bharat, highlighting the significant contributions of women scientists and innovators in science, technology, engineering, and mathematics. This theme celebrates women's role in driving research excellence, fostering gender inclusion in scientific fields, and empowering future generations of women and girls to pursue science and innovation for a developed and resilient India. As you know, National Science Day is not only to commemorate a historic scientific breakthrough, but also reinforce the importance of scientific thinking, critical curiosity, and equity in science as foundational pillars for national progress. With this very brief background of this day, I welcome our director, CSIR-NEERI, Dr. S. Venkatamohan, for his welcome remarks on this occasion. Sir, please.
Our chief guest, Professor Uma Ramakrishnan, National Center for Biological Science, Bangalore. Prakash Kumbare, my dear colleagues, students, and invitees. So good morning to everyone. On behalf of CSIR-NEERI, I'm very happy to welcome you all to this National Science Day celebration 2026. So every year, as you all know, on 28th February, we try to celebrate National Science Day for the discovery of Raman effect by Professor C.V. Raman in 1928. The discovery reflected that light changes in wavelength when scattered by molecule. This has led to many applications subsequently. This also earned first Nobel Prize in Science to India and also proved that world-class research can emerge from Indian laboratories. This Raman effect laid the foundation for modern spectroscopy and supports medical diagnostics, material science research, space research, environmental research. Specifically in environmental research, even though it started with fundamental physics, Raman spectroscopy supports environmental surveillance and monitoring in diverse ways, like detecting atmospheric pollutants and greenhouse gas, analyzing particulate matter, monitoring microplastics in wastewater, supporting carbon capture and climate research, apart from assessing hazardous material in environmental matrix. This laser-based Raman system are used globally for real-time air quality and aerosol profile. So from understanding molecules to protecting ecosystems, this journey reflects the evolution of Indian science in global context. So this year's theme for National Science Day announced by Department of Science and Technology. Women in Science Catalyzing Viksit Bharat. Across India, women scientists are leading breakthroughs in STEM, particularly specific to our domain like environmental biotechnology, climate modeling, sustainable materials, energy systems, AI-driven research. Leading as leaders, policy advisors, enterprises, and mentors. Our own Director General, Dr. Kalai Selvi, who is the first woman DZ, is leading CSIR as well as DSR in a very exemplary way. Many women directors and heads in CSS systems are leading labs and research centers. The participation of women scientists in STEM is critical and central. So in this context, it's pertinent to say a few of the names in this occasion who received conferred with Padma Shri Award, particularly in science, women scientists in last 2 years. Recently this year in 2026, Dr. Shubha Iyengar from CSIR National Aerospace Laboratory conferred with award for her outstanding work on improving aviation operations in extreme atmospheric conditions. Last year, two of the scientists, Dr. Neeraj Butler, who worked on women's health, particularly cervical cancer prevention, Professor Sonia Nityanand, whose work has been significant in immunohematology and sustained contribution to medical education, was recognized with the coveted Padma Shri Award. Several Padma Shri recipients supports community empowerment also, particularly in our domain, environmental domain, who supports environment sustainably. We try to see few of the names during this occasion, particularly under the theme today's science day theme, this year's science day theme. Chami Murmur, she was recognized for afforestation efforts and women's empowerment. Sala Maddal Timmakka from Karnataka transferred barren land into green corridor by nurturing hundreds of banyan trees. Tulasi Gowda, known as Encyclopedia of Forest, embodying indigenous ecological wisdom. Vandana Shiva, a champion of biodiversity conservation and sustainable agriculture. Rahibai Soma Popper, seed mother, preserving indigenous seed varieties. Purnima Devi Varman for her work in protecting endangered species through community-based conservation. These lives inspire, their lives inspire and remind the sustainability and social responsibility go hand to hand and thrive to communities, forests, and fields. So today our chief guest, Professor Uma Ramakrishnan, a very renowned and celebrated molecular ecologist. Professor Uma's pioneering work blends population genetics, evolutionary biology, and field ecology to understand how species respond to environmental change and human impact, with a strong emphasis on conserving India's unique wildlife and biodiversity. PhD from University of California, San Diego, and postdoctoral training at Stanford University. She is a fellow of Indian National Science Academy, with a lot of prestigious award, to name one, Parker Genter Conservation Award. I would like to thank Professor Uma, our chief guest, for taking time in her busy schedule to grace this occasion. Thank you, Professor Uma. And once again, a warm welcome to you all. Thank you very much for your attention.
Thank you, sir, for your welcome address and very nicely sharing of the insights about some of these pioneering women who have contributed a lot in science and environment, and also women from different walks of life who have conserved ecosystems and diversity of this country. I would request sir to please welcome Professor Ramakrishnan to the staff meeting, please. Thank you. Sir has briefly introduced our esteemed guest, Professor Ramakrishnan, but it's a pleasure for me as well to elaborate a little bit more on her work. As Sir has already mentioned, she is a leading molecular ecologist, an evolutionary biologist, a professor at NCBS. Her work sits at the interface of ecology, evolution, conservation, genetics, with a strong focus on understanding how species respond to environmental change across Indian subcontinent. Over the past 2 decades, she has built an internationally recognized research program that combines field ecology with cutting-edge molecular tools to address questions of population structure, connectivity, adaptation, and speciation. Dr. Ramakrishnan is especially well known for pioneering the use of non-invasive genetic sampling to study the wild tiger population in India. And beyond tigers, I should mention her work spans a diversity of taxa and ecosystems, including studies of speciation and diversification in the Western Ghats, one of the world's biodiversity hotspots. Her publications are in leading international journals. She has contributed fundamentally to conservation genomics that have also informed policy, management decisions, and even legal deliberations related to wildlife corridors and habitat protection. She has been conferred with multiple awards and recognitions, and that reflect her truly global standing in the field. She is also recipient of international awards and has multiple honors acknowledging both her research contributions and leadership in Indian science. With this very brief introduction, I know she is already innovating, and with her interdisciplinary work, she continues to shape how we understand biodiversity, evolution, and conservation in rapidly changing landscapes. She does give a lot of popular science lectures across the country and across the world. It's a pleasure to have her with us. So please join me in welcoming Professor Ramakrishnan to this audience. Thank you.
If it's there, if it's not, it's fine. No, I can see that. It's so great to see so many of you here for this talk. Thank you for the invitation. And hopefully I will not bore you too much. As Dr. Shalini said, I give a lot of public talks. So hopefully you haven't heard the stuff I'm going to talk about. Yeah.
Color mic.
Because I
you
walk
get
around a
that.
lot.
So that will be good.
Yeah. Yeah. Slide share. Here. I'll be like a news reporter. Yeah. Okay. Hi, everyone. Thank you so much for coming today. My name is Uma Ramakrishnan. Yeah. It. Light off Yeah. So my name is Uma Ramakrishnan, and I love this illustration. It's actually by a really talented cartoonist. And like I said, because I work on tigers, I've been lucky to have a lot of people who are kind of drawn by that hook, of tigers, because they're so beautiful. And I've just been basking in the orange glory of tigers for a large part of my career. So I thought it's really nice to think about, what. Should I just use this? Yeah. So basically, I thought, when it's of course, it's amazing that in this country we have a science day, right? And in our constitution, there is a line which says we are entitled to curiosity, to do research, to understand more about our world, and also codes us to have a scientific attitude. So that's really something very special in our constitution, right? The value of science is there, written in the constitution. And like all of us, I think we started our interest in science just from curiosity or some kind of a wonder. That's what science represents, right? And basically, even for me, when I look at these natural systems, this is a picture from Kanha. This is a leopard in Taroba, actually. You wonder, right? For millions of years, these systems have evolved. And around us, there's amazing diversity. And I basically am very frank about it. I really love animals. That's how I got interested in doing science. I just love animals, very curious. How do they think? How do they do these things? And can we understand them better? I also love genetics because it's actually quite quantitative and very it's very precise in some sense, at least Mendelian genetics, right? So it's very simple in that sense. And basically the idea of studying all of these species, and one of the best parts of science is working in teams. Science is not an individualistic activity. Unfortunately, these awards and stuff are to individuals, but overall, I think all of us as scientists, we love working in our labs. We love working with all of our early career researchers, students, postdocs, and that's basically part of the motivation. So I guess I think it's really critical for us to think what can we actually learn from nature and how can we better understand India and its biodiversity. So for me, it's a very personal, also almost, how should I say, patriotic journey because I really want to understand this biodiversity that surrounds us and what can we actually learn, which may be potentially useful for science as well as human well-being. So, of course, I think it's really important to talk about the ethos of science. How do we do science? I think it's critical that it has to be inclusive, participatory. We have to think of all kinds of people who are part of this process, and of course, think of capacity in general. So, I think science is not just about curiosity, but also a culture which we kind of promote of how we study species and interactions. So very briefly, my journey, like I said, I always loved animals and want to understand them better. But when I was young, I mean, I'm much older now. You guys have much younger students today have much more opportunities for interdisciplinary education, like in the ISERs and so on. But when I was doing my bachelor's, I had to basically choose: are you going to do chemistry, botany, zoology, and drop math, or are you going to do physics, chemistry, math, and retain it? And I got good guidance early on that if you're interested in ecology, it's a very quantitative science, better not to drop math, right? Plus, there was another additional problem. I couldn't draw. So, I keep seeing those people in botany and zoology drawing all of those leaves and all. And I was like, okay, there's no way I can do that. So let me just stick to doing more quantitative work. So I did this bachelor's in physics, chemistry, and math. But then the only biology master's I was eligible for was MSc Biotechnology. That was the only master's at that time which allowed non-biology students to also appear for the test. I got really lucky somehow. National entrance test. I mean, I'm not a good student like that at cracking exams, but somehow I got it. And basically, I was finished my master's, and I really thought I want to use genetics. That's how I got exposed to genetics to understand animals. Because I had been to forests, I'd watched, for example, elephants, and I really wondered what is going on in their lives. And then I thought, how much ever I watch them, I'm not going to be able to understand it. I need some kind of a cheat, some kind of a lens, which will give me more information about their lives. And at that time, genetics seemed like a really interesting kind of way to do that. So basically, I went to do my PhD in the US because there was really no one working on that area at that time in India. I tried to do some work at CCMB. As an intern, I worked in CCMB with Dr. Lalji Singh, but I knew he was doing a lot of work on DNA fingerprinting and so on, but they were working with blood samples. And I knew that if I wanted to work on wildlife, I had to use non-invasive samples. That is poop or saliva or hair. There is no way I was going to get blood samples from elephants to be able to do the kind of work, population level work. That is not just who is an elephant or how different is an elephant from something else, but populations of elephants, right? Different elephants. How different are elephants in South India versus somewhere else? For those kinds of questions, I had to have a broader sampling, more samples. I needed to work with non-invasive methods. And so even at that time, I went to Lalji Singh's lab and I said, can I try to extract DNA from some dung, some poop? So he said, okay, there's a colony of, where there's a lot of buffaloes. You can go and try and collect some buffalo dung. And I did it. I was the laughing stock of CCMB at the time. They were like, what is she doing collecting poop and bringing it into the lab? And it, of course, failed. It didn't work, right? That's another very important lesson, or not lesson, I don't want to sound preachy. There's just a lot of failure in science, but you have to keep trying, right? So anyway, so then I thought, okay, I'm going to try and learn these methods of population genetics Basically, that's understanding genetics of populations. But I'm focused on wildlife. I want to understand these rare and endangered species. So I need to be able to work with this bad quality material. And then I went in, I did a PhD and a postdoc, and I'll talk a little bit more about that. But then while I was in the US, I was so restless because I just wanted to come back and work on Indian landscapes, Indian species. While I was there, for example, I got opportunities to work on whales, to work on these endangered species called kangaroo rats. They're just this big, and I was like, what is this? So boring. I don't want to work on these species. I want to work on what I'm close to my heart, my landscapes. And so basically, I have to say, because there's so many students here, I had an incredibly bad PhD. The reason it was so bad, this is a very bad, sorry, pixelated picture. That's me from 25 years ago. And I was collecting elephant dung. I was working with Professor Sukumar. I wanted to look at paternity in elephants, makhnas, which are tuskless males versus tuskers, who is able to mate with the females and so on. But then India tested nuclear weapons. So that was 1996, right? India tested nuclear weapons and US declared sanctions on India. And so all projects, no students in US were allowed to work on projects in India. So there I had gone hoping to learn this genetics, use it as a tool to work on elephants in India, but then it was not possible. So then I had to struggle, figure out something else to do. And that's when I worked with the National Marine Fisheries Service on whales and dolphins. So I saw firsthand, National Marine Fisheries Service is like our forest department, but focused on marine and freshwater systems. And I saw how there was so much of science being infused into all these management decisions. And I was really hooked. That it's not just, finding out some theoretical, conceptually wonderful things, but it can actually be applied in the context of real conservation. But then I actually got lucky because I did a postdoc at Stanford, as Sir mentioned. But the amazing thing was the environment. So that's when I learned something about myself when I'm in a very toxic environment. Nowadays, we use the word toxic. That wasn't a word back then. But toxic environment, I don't feel like working. I don't like it when people are very mean and, not nice to you. That's one way you can encourage someone is to put them down. But for me, that doesn't encourage me. I like to work in places where people are friendly, open, and try to inspire you, right? They make you work or you end up working because you're inspired by them. They're so amazing and their attitude is so nice, all of those things. So I was really lucky to work with some amazing scientists. So this is Liz Hadley, and she's a paleoecologist. So she basically was actually a wildlife manager. She was in the US Fish and Wildlife Service as a manager in Yellowstone National Park. And then she was very keen to understand. So at that time, there were no wolves. Yellowstone National Park is a park in the US, one of the oldest, it is the oldest national park. There were no wolves there. And there was a talk of introducing wolves. And she was a paleontologist. So she said, let me find some fossil deposits. Let me look and see, were there ever wolves there? So we have our past with us, right? We seem often very preoccupied about the future, but the past can also tell us about the future, right? So she found that in fossil deposits, there were wolves there. And so it was kind of a go-ahead for the government to introduce wolves in Yellowstone National Park. So she was so excited about this, discovery and stuff. She later became a professor. She did a PhD and she was a professor at Stanford. And with her, I worked on ancient DNA, looking at the effect of climate change in the last 10,000 years on animals. So that was really fun because, like I said here, I had a terrible PhD. I felt I was really stupid. I was very low in confidence. And then when I went to this other environment, Stanford is a much more competitive, you would say, like compared to UC San Diego, it's a much more high pressure place, and it was high pressure, but the people were very nice and they were very inspiring. So that helped me, and I suddenly felt like after so many years of study, and I felt like I was really stupid, I suddenly felt like my brain has opened up. I'm thinking about ideas, and I really had an amazing time. So that was great. And then I came back to India because I told you I was desperate to come back to India. And it was a huge transition. So this is probably I'm giving you a slightly more personal talk. I hope that's okay. But basically, I had a daughter also. I came back, I had a daughter, I started a job. And I knew nothing about how to be a scientist. And as scientists, we are supposed to be managers. We have to run labs, have students, raise money, submit UCs. We don't have any training in any of that, right? We are just learning science, and then now like here's your lab, buy the equipment, figure out how to run it. And really, I had no clue. I was completely clueless. But somehow struggled through, and I was also very lucky at that time to get a call from a famous carnivore ecologist called Ullas Kadam, who just called me and he said, I've been studying tigers and their ecology. Would you be interested in studying their genetics? Right? And that was very nice of him because he was a very established scientist. I was nobody. I had just landed up, right? And we just had a conversation. We said, okay, let's do it. And then again, I was lucky because I got to work. The first student who worked with me and him was a guy called Anish, who later went on to found the Wildlife Conservation Trust. So we have a wonderful program in NCBS called the Master's Program in Wildlife Biology and Conservation. And many of the students who come to this program are very motivated. And it's a joy and really a learning experience for us to work with them. So I did different kinds of work. I worked on estimating population size of tigers in a park, not using camera traps, but using genetics. How can we do this? So you just walk in the park, you see tiger poop. Any of you have been to a tiger reserve? No, there's a lot. You may not see a tiger, but tigers mark their territories by pooping, and you can see a lot of tiger poop, more than sometimes you see tigers, right? So the most abundant source of biological material is poop. So basically, if you bring that back to the lab, you identify individuals based on genetic differences, and then you use a mark recapture model. It's just a model where you say, how many times have I captured and recaptured an individual? You can probabilistically predict how many individuals are in that population. Now, the good thing about this study is it's really old now, 2009, but at the same time, they were also doing camera trapping. So that's the best way to start off a completely new kind of study. If you're going to start with only unknowns, then how do you know whether what you find is correct or not? But if you have some other data, so there's a camera trap data which is estimating at the same time as a completely independent genetic estimate, and the two numbers matched. So that suggested that it's not some garbage I'm getting out of this, because it's kind of indirect, right? I'm just getting some samples to the lab, doing something, who knows? Maybe I'm doing it wrong. Maybe there's contamination. So that was really rewarding, and that thrill is mind-blowing, right? That you just collected poop and you can actually estimate without seeing a single tiger, you can estimate the number of animals in that particular park. We did some other work which actually long ago suggested that tigers' populations have been through bottlenecks. And in fact, the timing of these bottlenecks, bottleneck means a crash, is coincident with when the British started bounty hunting. So there was probably a lot of hunting of tigers throughout. And I'm going to get back to this question again towards the end. But basically, there were some signals of when that actually started. So just to show you then, I had another daughter. So that was also tough. How to manage, all this work that's happening with two small kids. But like I said, I've been really lucky to have a lot of support to be able to continue to do kind of everything together. So that was the transition. I said a transition period when I first came back. And that was also what's called pre-tenure. So at least in the CSIR system, the first 6 years or so, you're kind of on trial. So basically, you have a job, but unless you perform well, you may not be made permanent or you may not be retained at the institution. So this is a practice which is quite common in several places around the world, but not in all institutions in India. But we had this practice, and I think it's good because the TIFR system also provides you with a lot of funding and support. So anyway, so that pre-tenure phase was quite difficult because it was a transition to doing research here. And at the same time, I really didn't know who I was. What am I doing, right? It was an idea of a fascination for science, but doing it practically is a different thing. And then basically, when for the next 5 or so years, I don't think I was thinking too much. A lot of good people joined the lab because, like was mentioned earlier, a unique lab. So a lot of really talented students and postdocs joined the lab. For example, this is Robin, and we worked, for example, with him on understanding speciation in the Western Ghats. And this is really interesting because we actually showed that for all species, the biggest barrier in the Western Ghats is the Palghat Gap. So if you want to look for two individuals of a species which are most different from each other, they will always be on either side of the Palghat Gap. So this is a specific place, that is the Western Ghats, but a very generalizable prediction, right? And we also showed, for example, that there is speciation or diversification in the Western Ghats driven by the shape of the mountain. So basically, when there's a very deep valley, you see species on either side of the valley. And this harks back to very dominant paradigms in evolutionary biology, that is allopatric speciation. Speciation or diversification, new species are created when two populations are separated. If they're mixing continuously, you won't have speciation. So because of the Palghat Gap, this deep valley, these two populations become different, and then they become different species. Now, this is also important for conservation, right? Because what you're saying is then the mountain has more species than a flat landscape, right? And basically, this led to actually a whole popular idea of, the term was there, sky islands, that especially these montane species which live in high elevations, live on islands in the sky, right? Because this valley is inhospitable for them. They can't live there. And so they live on these mountain tops, and over time, they become different. So, this was some work really initiated, or like the ideas initiated by Robin. I just allowed him space and intellectual discussion to go forward, right? And now, it's something which he's a little sad about, but in the recent Wildlife Protection Act, what was originally thought of one species of shola kili and one species of laughing thrush is now 3 and 4, and they are all listed in Schedule 1 of the Wildlife Protection Act. So this is evolutionary biology research talking about the shape of a mountain, right? And how that leads to creation of biodiversity. But this creation of biodiversity, the process, results in species with small distributions, right? That is, they have small areas where they live, and that makes them endangered, right? And that then requires protection, okay? So, of course, there are some, we can argue about the effect of the Wildlife Protection Act. It's more kind of punishing than conservation oriented. We can talk about that if anyone has questions. But it's really cool that this actually, just scientific research, resulted in these species being recognized for conservation and protection. But like I said, I was still not thinking too much. But I think at that point I started thinking, okay, now I have finished 10 or so to 12 years of my career. I need to really think of what I want to do. What is my interest? Let me get back to that. That's the core. So basically, I'll talk to you a little bit today about tiger population genetics and how it can inform conservation, and then maybe a little bit about the ongoing work, which to me is really, really exciting. So there's a difference between, okay, so also around this time, there was a revolution happening in genetics, okay? So we started having this new technology. So basically the original way to look at DNA sequence was what is called Sanger sequencing, where you read a base pair one by one. So DNA is just a long spelling, right? With only 4 letters, but very, very long for the whole genome. So you can read it one by one, and that takes quite some time. So the first whole genome, for example, for humans, took 10 years and 10 institutions across the world, okay, to put it together, because they read it bit by bit. But now there was this new technology which allowed you to read it in parallel. So you cut up this long book into small pieces and read them all in parallel. So you can put together the whole book very quickly by just reading everything in parallel. This was called next generation sequencing. Okay. And we encountered next generation sequencing, for example, during COVID, where there was a constant report of the new lineages and so on. All of that was based on next generation sequencing the coronavirus, right? So basically, all of this was happening, but still, it wasn't very applicable to, wild species, endangered species, and so on. But the idea is simple. Imagine the difference between genetics and genomics, right? So genetics is, imagine you have a snow leopard, you have many pieces of the puzzle. The whole genome is positional as well as picture information, okay? For example. But many times what we study are just markers, like we just take 3 pieces of the puzzle and we look at that. You can see that depending on which piece of the puzzle you take, those 3 pieces may be identical, right, in how they look. But if you look at the entire puzzle, you have much more information or power, okay? So basically, the idea for going for genomics is not a megalomaniacal thing. It's not just because you want more data, but that data is actually statistically better. It has more power for you to be able to tell differences between individuals and so on and so forth. Okay. So, but then we have to have a reference or some kind of a framework. Suppose I go and start sequencing tiger genomes. How do I say this is variable, that is not variable? I need to have some reference to compare it to. And there was no reference genome for tigers in India. Okay. So what we did is we created a tiger genome reference. This was based on a famous individual from Ranthambore called Pachli. And she's a founder of Ranthambore. But basically what we did is we sequenced this genome in various ways and put together a good framework, like a scale, which we sequence anything else, we can compare it to. Okay. So this was just in some sense, it's just a baseline. It doesn't tell you anything very exciting, but without it, you cannot go forward. And because tigers are a species we continue to work on, not just me, but many others, this is there for them to use for perpetuity. We also did some sampling across the whole range of tigers. So you can see here, but basically you can see we sampled tigers and tiger genomes from across India, Southeast Asia, Russian Far East, and so on. And you can see, for example, in this PCA plot, that that's just a statistical way of separating things which are different, right? It's got no assumptions about genetics. You can see that, Indian tigers cluster together, tigers from the Russian Far East cluster together, Malayan and Sumatran tigers. What's really interesting is this plot here, which is a rarefaction analysis and shows genetic diversity as a function of sample size. And you can see that by far, Indian tigers, which are in pink, have really high genetic diversity together. All Indian tigers together have very high genetic diversity. And if you calculate the numbers, something like 80% of global tiger genetic diversity is in India. Now, that is really important because it's not just that we should conserve tigers because they're a national animal or they're beautiful, but India has a responsibility to conserve tigers because most of the future evolution potential of tigers is in India. So this was a really interesting study for us to know that it's important to also conserve these relationships. We did multiple other studies, for example, as Sir mentioned, looking at connectivity within central India. And this helped, for example, was one of the studies used in court to petition for an overpass for wildlife, for cars, underpass for wildlife. And I don't know whether you guys have seen it. It's on the in the page on the road to NH7. But we also then found 2 populations of tigers which were isolated. And I'll briefly talk about those 2. So why is isolation bad? Why is it bad to have small isolated populations? Well, what happens in small isolated populations is everybody's related. And then you have what is called inbreeding. Inbreeding is mating between relatives. But why is inbreeding bad? So inbreeding is bad because all relatives have the same bad variation. Not all variation is good. Some variation could be bad. but it's just hiding in our genomes because it's not expressed, right? But when individuals are related, they have the same bad variation, and as a result, you have the expression of this bad variation, and that is called inbreeding depression. So this is theory, right? We've seen inbreeding depression in many species, but we've not seen it in Indian tigers. Let me be very frank. What we were trying to assess first of all is there inbreeding? Is there mating between relatives, which results in low genetic variation, high homozygosity, and also these bad mutations? So we basically did this study several years ago in Ranthambore, and the tigers there, they are isolated. And what we showed is we sequenced 48 whole genomes from across India. And what we showed is that tigers in Ranthambore are twice as inbred as those in any other location in India. And we also see that inbred individuals have this potentially bad variation. And there's also, though, a signature of what's called purging. That is the very bad mutation. As soon as there's inbreeding, those individuals which have it, they just die, right? So there's a suggestion statistically that there has been some purging or removal of these very bad mutations. But see, this concept of inbreeding using genomics is not how we understand it as in school, right? Inbreeding is about pedigrees, that is family trees and relationships. So for the past 10 years, we've been trying to create a family tree in Ranthambore. What do we do? Every year we go out, we follow mothers and their cubs, and we sample the hair where they've been sitting. So we take pictures because after they get up, it's sometimes very confusing, right? So when they sit down, we take pictures, or they're next to this tree, next to that, etc. When they move away, we look, and you can see we're looking over there. We sample hair. You can hardly see the hair. It's like, but they actually, we never looked before. There's a lot of hair where tigers sit. Lots of hair, like 200, 300 hair. And then we extract the DNA and we sequence genomes. So this is the potential family tree of Ranthambore. It was supposedly founded by 9 females. Okay, so you can see, for example, the females and their offspring are known, but the fathers are not known, right? So the idea is, can we figure out from this DNA the paternity and the entire pedigree? So first of all, I told you there was this, by the way, this is not yet published, it's still something we're working on. I told you that there were 9 females which founded Ranthambore, right? There were 9 matrilines. But when we look at mitochondrial DNA, which is basically passed on from your mother, we see there's only basically two matrilines. So those 9 females were actually like sets of 2 sisters, okay? 2 sets of females, others were sisters. Okay, so basically the founding event itself has now shrunk. Right? So here's the pedigree. It's a little bit messy, but what you can see over here is that there are the mate pairs, which is the square is a male, right, and the circle is a female. The mate pairs which have darker colors suggest that the mate pairs are related. So what we found is that overall there isn't mating between relatives, but there is mating between uncle and niece, aunt and nephew, and half-sibs. So there's no first-degree relative mating. That is, sister and brother, mother and son. There's no such mating, such inbreeding, but there is evidence of half-sib or the second-degree relative mating. What was also interesting is if we look at inbreeding or heterozygosity, lack of heterozygosity as a function of birth year, what we see is that it's definitely increasing. So despite the fact that we don't yet see inbreeding depression, Ranthambore is isolated. It was founded by very few individuals, and over time, the amount of inbreeding is increasing, right? So this makes you also, of course, wonder, what is the effect of this? See, you're going on saying things like genetic variation is good, we should have more variation, the bad variation is bad, but why are we not seeing it? So this is something which is much harder to answer, but we know, for example, there is some females who have not bred for 6 years, right? So maybe we are not able to see that inbreeding depression. Maybe they're not getting pregnant because they're inbred, or they're having miscarriages because those embryos have a lot of mutations. We're not able to see that, right? So we still need more time. And already, for example, I'm sure, the forest department would be asking us, you've been studying for 10 years and you still don't have answers. So that's unfortunately the truth. It takes sometimes very long time for us to answer these questions. So very quickly, another population which has these black tigers in Odisha, and we were able to find the mutation which causes this black phenotype or the way they look this way. And it turns out that basically this population also has very low genetic variation and potential for inbreeding. So we had suggested to the Odisha Forest Department it would be good to bring individuals from somewhere else to supplement or enhance the gene pool in Simlipal, in Odisha. But the question then was, where should we bring these individuals from? Fine, bring individuals, but from where? So we developed this novel framework where we said, okay, let's consider 3 factors. One is the genetic diversity itself. Wherever you bring individuals from, you want them to have high genetic variation, so they're actually bringing in something and not just an animal. The second is how much of compatibility is there. That is, if they have the same bad mutations as they're there in Simlipal, then it really doesn't make much difference. It doesn't alleviate inbreeding in that sense. So anyway, so I'll just cut a long story short. We did these analyses, and based on these analyses, we suggested that Tadoba would be a good source for individuals to Simlipal. Based on this and many other logistic considerations, the Odisha Forest Department decided to do this, and we basically developed some kind of a flow chart, and we're trying to now work on making this more, say, manager friendly. How do managers, this is our perspective, the managers may have a different perspective, but how do we provide evidence? We talked about evidence initially, such that they can make decisions to execute these kind of genetic rescue or supplementation processes. And basically, as you probably have heard, 2 tigresses were introduced, and now we have a running experiment. Once these tigresses get pregnant, they have offspring, we will see how the infusion of genetic variation will change this population. So it's a natural experiment. So we don't do experiments in the lab. We do extraction of DNA and stuff, but these kind of experiments are a way to continue studying. So again, we have to continue studying this population. If we stop, then we really won't know whether what we did was right or wrong. And it's very important for us to have that data going forward. So very quickly, you all have heard a lot about human-wildlife conflict, where there's a lot of killing of course, livestock, but also humans. And so this is something which we were interested in. I had a very talented student called Himanshu Chaitani, and he was very keen to know whether from these killed animals, we can actually sample something which will tell us about which individual. Like I said, we can go and collect dung and identify feces and identify individuals. So can we actually do this from that dead cattle? Can we sample something and figure out who was the individual involved? So basically, the short answer is yes. We go to a kill site. There are lick marks. Cats always lick their prey. There's a lot of hair of the predator. There's also jugular bite marks. So we can just take like a swab and sample there. We can bring it back to the lab. We can identify the individual who was involved. And the success rate was pretty good. At about 83% of kill sites, we did this outside of Bandhavgarh mostly and a little bit outside of Kanha, we were able to identify with this trace DNA, it's like forensics, right? With this trace DNA, we can identify who the predator is, which is pretty good, right? Unfortunately, this doesn't really help because we have the DNA of the predator, but we don't know physically who is that animal, right? So for that, we need to have a database, a geogenetic database where we have an animal and we have its genetic signature. And so then we can actually, when we have a kill site, we can actually match it to figure out which individual was involved. Now, these kind of efforts will require immense collaboration between forest department and scientists. And I do hope that we can move there in the future. There's also now new technologies. There's a sequencer called a nanopore sequencer, which is just a handheld sequencer. So we could actually plan to do this in the field. The other problem is time. science is very slow. Managers require very quick answers, right? So if I take a sample, it goes to the lab and the whole process is slow. But if we can actually do this in the field, and there are people doing it with other species globally, like with leopards and other endangered species, leopards in Africa, we can actually quickly give a response. So those kinds of things, I mean, especially organizations like NEERI, be really exciting to take up those kind of applied initiatives. You could also build these individual identification panels, have standardized approaches, which is continuously checking to make sure the data is reliable and so on. What I'm saying is, it's also possible to develop kind of a tool and an application going forward. Okay, so I talked mainly about tigers, but clearly tigers are one species. There are many endangered species, and I have a really talented student who's developed again these panels of markers which work for all cats. So this is great. It's called Philiplex. I go to a landscape, I collect a sample, and I can then do multi-species connectivity. How are the different species, tigers, leopards, jungle cats, all kinds of species, cats, not all species, but all cats connected in a landscape. She's just finishing her PhD and she's done some beautiful work in the Tarai arc, and I'll just point out this for you. So basically, there are these 4 parts. This is Katarnia Ghat, Dudhwa, Pilibhit, and Kishanpur. Okay, and you have, based on genetic data, she collected about 3,000 samples, fecal samples, walking more than 8,000 kilometers. This is no easy task, let me tell you. A lot of work. And then she was able to get data for fishing cat, jungle cat, sloth bear, leopard cat, and tiger. And what you're seeing is the connectivity maps. That is how connected between the protected areas these different species are. What's interesting is fishing cat, which is very dependent on water, is very well connected across the 4 because this area has a lot of rivers. Basically, Tarai is a floodplain, right? It was a boggy kind of environment. But on the other hand, if you see, by the way, outside of these parks is very hard edge, sugarcane, dense agriculture, right? And jungle cat and leopard, these two, the pink and the blue, seem to be fine with the agriculture. They seem to be fine with sugarcane. They are having connectivity through those 4 areas despite the sugarcane. But if you look at tiger and sloth bear, this connectivity is breaking down, especially here between Dudhwa and Katarnia Ghat. In fact, to her extensive surveys, she never found sloth bear in Katarnia Ghat, suggesting that they're potentially extinct there, driven by this loss of connectivity. Okay, we can't be sure, but potentially. So now you'll probably say, so what? How does this matter? It matters because what it suggests is this connection is extremely important to maintain for the future. And the other thing she did is, she said, suppose we take tiger corridors and say, how well do tiger corridors work for other species? The answer is only 40% of corridors for other species are covered by tigers. So tiger cannot be representative for everyone, right? So this study actually shows that, and it's actually kind of actionable, right? That, this is very fresh. We've not yet gone back to the forest department and discussed it, but I hope we can, and then they can also think of, it's one thing to say, please try and protect that area. How they do it on the ground, that is of course the job of managers, but hopefully this kind of science will aid in their ability to do so. So, of course, as was mentioned earlier, we've been working for some time with people in Taroba Andheri Tiger Reserve on understanding diet. And we're actually finally at a point where we're doing something really interesting. We have gotten the diet from the metabarcoding, that is the fecal sample has tiger DNA, but it also has what it ate, the prey, right? So we can look at diet across the core and the buffer and the territorial, and we can also look at prey density in the core and the territorial in the buffer. And then we can try to relate those two and ask, are they eating what is abundant or are they picking something else? And I don't have those results to show you here, but very preliminary results show that in the core, tigers are preferring medium-sized prey, which is cheetal and sambar and so on. But in the buffer, they're actually avoiding them because they are preferring cattle. Okay? Not entirely, but preference-wise in terms of compared to the abundance. So this is something which is ongoing, and these kinds of data will also provide very interesting insights on ecology. So we're using genetic tools here to understand ecology. So another very exciting project, and I'll wrap up soon, I'm sorry if I'm taking too long, is trying to understand how we got here, right? So these are some amazing paintings. This is a painting from the Mughal times of a tiger hunt. And of course, these are very famous pictures or, visuals of British Raj and hunting. There was immense hunting of tigers, right? And that is why, so tigers are not a very specialized species which can only live in a very specific kind of forest. They live from tundra all the way to rainforest. They are very generalist, not a generalist, but they can live in many, very adaptable. They can live in many environments. So their rarity has been driven by, human hunting and habitat fragmentation, right? So now the question is, which was it? I said it in the same breath, human hunting and habitat fragmentation. Which was it? Can we figure out what actually led over time to the endangerment of tigers? So what are we doing for this? We're actually going back in time and we're using these old skins and skulls which people hunted. So this is really an amazing journey. We've been collecting samples for the last 3 or so years. This is a picture from Chhattisgarh, basically a place called Akaltara. This is my student in the UK at the Bristol Museum, where they have a lot of skulls, and she learned how to drill and get this bone powder and extract DNA from it. This is from the BNHS. It turns out that, the nose has a lot of very thin bones. They're called turbinals, and you can just put your forceps and pull them out. They have a lot of DNA because there was a lot of, blood over there. And this is a picture of me risking my life, I think, standing on this really rickety ladder in the Sarguja Maharaja's palace, which you can see was completely burnt. Look at the quality of this trophy. It's really bad, right? But after so much of effort, and this is how we have to work, we set up a special lab for this. We have to wear full PPE. Why? Not because we are going to get some pathogens, but we don't want our DNA to go into those samples, right? And basically, now we have 250 plus, so about 360 samples from across India. Unfortunately, we were not able to collect samples from Maharashtra because we didn't get the permits. But basically, we're focusing on this landscape, that is Rajasthan, MP, Chhattisgarh, Jharkhand, Odisha, West Bengal, right? And what's very interesting so far is that if you look at the amount of DNA which different samples gave, the median concentration of DNA from the Indian trophies was 0.24, right, nanogram per microliter, but the UK ones gave 200% more. The reason is because they were preserved well. They were kept in museums in temperature control, whereas our trophies are sitting in people's houses and sweating it out, basically, right? But still, that said, nobody has attempted it. And I'm so happy. I hope that, those animals that died in vain, maybe they get a story. They get to tell their story of their past. So this is ongoing work, which we're trying to actually sequence genomes from all of these samples, compare them to modern data and ask, was it hunting or habitat fragmentation, or how much did both contribute to the isolation and breakup of these tiger populations across the country? So maybe this is purely academic, I don't know, but I hope it will also help inform strategies. For example, if you want to rewild Chhattisgarh with tigers, where should they be from? This might tell us about the natural dispersals or connectivities of historical populations, which may help us think about in the future, how do we go forward with these areas where tigers are currently extinct. Okay, this is my last slide, but I have been really lucky to get many opportunities in my life. And like I said, I worked on biogeography or understanding which species is where and why, the speciation in the Western Ghats and so on. And basically, these species are communities, right? You have communities of rodents, communities of bats, communities of birds, but on top of those are the co-occurring microbes which live with them. We all know bats harbor a lot of viruses, and they can do that. They're somehow tolerant of viruses and bacteria because they have a suppressed immune system. So bats, they're so tiny, and usually lifespan correlates with body size. If you're small, you die soon. But bats have conquered aging, and small bats, few less than 100 grams, live for 5-10 years. It's really cool. So they're very interesting, but because of that, that's interesting, but they become hazardous potentially to us because they may transmit these zoonoses or spillover events from wildlife to humans. So we got this really great opportunity funded by the DBT Northeast program to work in Northeast India with some amazing partners. We have here public health specialists, doctors, genomicists, wildlife ecologists. And what we're doing is we're sampling rodents and bats. You can see the fieldwork is very hard. For the bats, we have to work at night and basically put up these traps outside caves, which they fly into, and we sample them, rodents as well. And we are basically trying to look at the sharing of pathogens between rodents, bats, livestock, and humans. So basically, the goal is that each of our study sites is a village, and in the village we sample livestock and humans, and then in the surrounding forest, cave, jhum agriculture, which is slash and burn, we sample bats and rodents. And what we hope to do is come up with a measure of risk. So the idea is that, like I said, the hazard is there, bats are there, and in places like Meghalaya, there are these limestone cave systems. Some caves have 15 species of bats, huge number of bats, right? So clearly, the evolution of pathogens will be higher there. There are more species, and there's a lot of cross spillover and so on. So there might be higher hazard in some places, right? That is the fact that there are many species of bats and rodents, and they all potentially carry pathogens. That's just the hazard. But ultimately, spillover is determined by exposure. That is, how often are the humans or livestock interacting with those bats, interacting with those rodents, right? That could be through hunting and consumption. It could be through just, interaction somehow. So for this, we are actually doing a lot of human surveys, community surveys to understand interactions with wildlife. But we're also collaborating with a chemical engineer, Rahul Roy at IISc, who's developed serology. So we can actually measure whether the bats, humans, and livestock share a common immune response. Have they been exposed to the same pathogen, which would show that they're sharing that pathogen, right? And finally, there's vulnerability. For example, some places we're working in Nagaland, no doctor has been there for the last decade. It's such a remote place, right? So they have high vulnerability compared to, say, a well-connected village. So that's again a socio-economic thing. But our goal is to try and plot this risk across about 20 districts in the Northeast, Arunachal, Nagaland, and Meghalaya. We just started this work about 8 months ago. We already have about 400 samples from bats and rodents of about 35 species. We already got new records for India because nobody has studied there. It's not anything great. It's just that nobody studied there, right? Nobody has gone and looked at these species there. So I'm sure this will basically lead to a lot of interesting work. But more than that, I hope it kicks open the door that when we think about zoonosis, we should not think only about livestock and humans. Wildlife and ecology is the driver. So when we think of, say, you think of coronavirus, the pandemic, what happened? It was a response, vaccination, etc. This is all response after the fact. But how about preparedness? How about understanding what types of scenarios actually result in spillover? So that's the hope. I don't know whether we'll succeed, maybe we'll fail, but anyway. So basically, just to come back to this timeline, this is where I am now, like done a lot of work and focused in the last, I think I should say, last 10 years, I've been a better scientist. I've focused more. But now I'm 53. I have like 7 years left before I retire. So I need to really think what I'm doing and how do I want to end this journey as a scientist? What is the consolidated work I want to do? I think it's good to, I'm not saying it in a bad way. Like it's really good. It's very easy to get busy and just do lots of urgent but not useful things. And it's very difficult to focus and concentrate and think about the meaningful things. So I'm hoping that I have the courage to do that in this last lap. And like I said, I've been lucky to have many opportunities for public engagement and also this really amazing opportunity to work with a photographer who's a world-class photographer and did this story in National Geographic, which was on the cover. And of course, I do nothing much, right? While I'm here, I have a fantastic team of people. Like I said, science is social. I'm really lucky to work with some amazing young people throughout my journey. And since it's a Women in Science talk, I have to, in the end, thank my family because they have been supportive of the poop in the fridge and the blood in the fridge and all of these things and explain to their classmates how their mother works on shit. So yeah, so thank you so much for your attention and I'm happy to take any questions. Thank you for the invitation. I hope it wasn't too long.
No,
Do we have questions?
Hello.
Yeah.
Very exciting and very interesting talk. Amit Bantiwal from NEERI.
Okay.
Just to, out of curiosity, although must be established, equally important and interesting to learn the impact of environment and pollution on the wildlife.
Yes.
So, and how we can integrate these findings into that. Can you throw more light on it?
Yeah, I think that's something I've thought about for many years now. I'm not sure how to do it. There's a lot of studies which show that there's bioaccumulation of toxins in the food chain. So clearly, apex predators like tigers and stuff will have an accumulation of toxins. So what is the impact of these toxins is something which is really interesting. It's been studied, for example, in the case of mining and bats and stuff in South America, and they've shown there's changes in methylation patterns and expression, but that requires good samples, which we don't have. Usually, we don't very easily get, but a targeted study on that would be really interesting to look at the impacts of even something like plastic. There was a study, it was a citizen science study, which suggested that, even for sometimes elephants in protected area, there's plastic in the dung. But what is the effect of these toxins? Well, we can't talk about effect initially, but how much of overlap is there, right, of these toxins with our wildlife? That itself is a very interesting. And then, of course, cows, my God, they eat all kinds of stuff, right? You see them around. And so if say, the individuals outside the protected area are consuming or are in more interface with these toxins, what is the effect on them? Yeah.
Thank you.
Tigers basically maintain population dynamics of certain species. So my question is whether that population dynamics maintenance is basically from genetics or it's coming from supreme consciousness of tigers.
Oh, so I think it's just ecology. There's these concepts in ecology of top-down versus bottom-up control of ecosystems, and top predators are supposed to exert top-down control of ecosystems. And there are several studies which show that if you remove the top predator, there's what's called mesopredator release. That is, the other predator starts taking over. And for example, what is really interesting is we see now there are a lot of our protected areas with conservation. Tigers have increased in abundance and have really become top predators. So if you see I didn't show those data, but the sampling in the Terai of the samples, half of them were tigers. So tigers are very big. Their abundance is not that high as is jungle cat, for example. But because they are dominant and they are also physically dominant, they tend to push species outside or they exist at lower densities. So they exert top-down control in terms of dominance, behavioral dominance, as well as controlling populations of prey. I hope that makes sense.
Hello.
Yeah.
Thank you, ma'am. It was very inspiring and informative talk. My question is, few years back, India brought cheetahs to Kuno National Park. So, time and time we were listening to the news that some cubs have been died and some cheetahs have died. And then again, we brought some more. So, is it related to somewhat in breeding and all?
So, I mean, it could be, but we can't say it is. Okay, until we study it, we can't say for sure. Generally, we know general scientific research suggests that the cheetahs are inbred in general. They have some sperm abnormalities and so on. I don't know anything about the specific cheetahs which were brought. And generally, it does take time for populations to establish. So it might have taken some time for them to, acclimatize to, basically the summers in India, for example, much hotter. There's a lot of differences, right? So I think that I can't say for sure, but it would definitely be an interesting study. Like I said, many of these are experiments, right? Introducing the cheetahs was an experiment. So it would be really interesting to actually study the impact of the cheetahs on the ecosystem. How did the other carnivores change? What are they eating? How are they avoiding or facilitating other carnivores? But for that, we need to have a scientific study ongoing with the management. So yeah, I'm not doing that. Maybe there are some others who are participating. I think there's a question.
Interesting. Since I was always curious about why the corridors are being made,
Yeah.
I always wondered, the animals do have the human intelligence to use the corridors only, just like we use the zebra crossing to cross the road? Is it something like that?
So basically the way we design corridors is not taking into account animal behavior. So what we are doing is we are looking at connectivity, say, between different areas, and we are assigning resistance. So if we don't see connectivity between two areas and there's lots of roads and lots of cities, we infer that these roads and cities are bad for connectivity. And so in this way, we infer what land use type, forest, road, city, agriculture, how does it facilitate or not facilitate movement? Then we look at the landscape and we model what is the best pathways for connectivity. And then we use that to say this is a corridor. But that is not taking into account tiger behavior or tiger decision making. How do tigers decide? Should I go here or there? And that is something much harder. We would need a lot of data from many radio-collared individuals and do models to think about it. But that has not been done. Mostly our connectivity is designed like this. Now, clearly, tigers are moving very long distances. So you might say, why do we even need these corridors? But those are very rare cases. I mean, of course, tigers are very charismatic. So you pick them up, the media picks it up. But effective movement is something without corridors. For example, in Central India, when we did models, the chances of extinction increase by 50% if all the protected areas are fenced and there's no movement outside of it.
You were talking about in the sample with the Karai region.
Yes.
Yeah, you showed that there were some gaps. I assume those gaps might be because of human habitation.
Yes.
So you mapped the animals and you found that some of the animals were found only in the area and they were not crossing that human habitation border. So how is that the animals, if they are not allowed to move across, they are able to cross over the concept of the bad repercussion of inbreeding?
No, it's not like that. No, there's no. Okay, so that was kind of going towards your question also. At least the way we do science, we don't assume some cognition in animals in this. They don't know when we also make decisions, we generally think of short-term effects. We are not thinking about long-term effects, right? So the tigers are not thinking about inbreeding. They are moving. Why do they move? When there's high density, individuals disperse, for example, to find new territories. And as they're dispersing, if they find a territory which looks, an area or a habitat which looks good, they may settle down. Now, one of the reasons why there may be these long distance movements is there is no place for them to, Mr. Kale would probably be much better able to answer this question. There may not be good enough habitat for them to settle and breed and so on, so they keep moving. Yes. Yeah, because that's at least what the Ranthambore pedigree also suggests, that they do know they're not mating with siblings, right? But in terms of, I'm not sure, unless we can go into the mind of a tiger, I don't think we can understand that motivation. All we can do is facilitate the landscape for movement. Now they could still not move, you know?
Like, I have another question, ma'am. Sorry for taking your time. Like, I'm a microbiologist, and in microbial ecology, it is well understood that when you introduce a foreign community, microbial community, like we humans think it is work best and we want to introduce in it. The native community always tries to remove it outshadow it. So how well it happens with the animals, like
Sure. So that's what I said. Like, for example, when these two tigresses were introduced from Tadoba, there was a huge, all kinds of things happened. They wandered off. They moved away, and it was difficult for them to get territories. So I'm not saying there aren't behavioral issues like this. There are. Our understanding of them is very poor. Like I was saying about, these tigers outside, they're eating all of this stuff. We have no clue, right? Because the question is, how do we study it? If you're doing a project or a scientific research, you have to have a tool to be able to answer the question. If you cannot, how can I know what the tiger is thinking, right? It's not possible. So
thank you so much, ma'am.
Yeah, I guess.
I'm Dr. Musira.
Yeah.
Basically, NEERI is involved in the AI studies.
Right.
So, as I understood, your work is like it will take a lot of time, right?
Right.
Three, 4 years.
Yes.
But in our AI studies, we have the time like 1 year, 6 months like that.
Yes.
So in that, we have to make the flora and fauna.
Yeah.
For fauna, my question is there. From your study, how we can take the help of the.
So it varies. Yeah, it varies depending on the taxa. But for example, for mammals, camera traps as well as environmental DNA sampling to look at biodiversity can be really useful. Same for insects also. Some kind of environmental DNA approaches from leaves and so on. And it would be really nice if you have access to experimental plots or some. I don't know whether you have some land or it could be somewhere like where you know what the biodiversity is. You could do an experiment where you actually do different kinds of sampling. And you look at what is the most effective and fastest way to capture the biodiversity best, right? So you do everything in that plot. You set up cameras, you use everything possible, eDNA, all kinds of things. And then you see, 5 days of eDNA sampling is equivalent to 10 days of camera tracking or something like that. So you can come up with a framework which will allow you to say, okay, in this, and then that will vary based on the diversity itself, right? As well as the size, right? So you'll have to set up some kind of an experimental framework to assess how much of sampling is enough in what way to get at the biodiversity. And that would be really great. Because otherwise it's very, ad hoc. I'm not saying that the EIAs that NEERI does this, but I'm saying in general it could be ad hoc. You could just say, look at expert opinion or something like that. For birds, it could be point counts. So it just varies depending on the species.
Thank you, ma'am.
One of your studies, you said you collected samples from the kills,
Yes.
like their hairs
Yeah,
or leaks, etc.
Yeah.
But as we all know, once the tiger makes a kill,
Yeah.
he will eat it for maybe 3-4 days,
Yes.
and normally he's stationed there near the kill.
Yeah.
So
Yeah.
whether you had any such,
Yeah,
that was very risky,
Very risky.
and how you managed those situations,
So,
what
like
precautions you took.
Like I said, the student was a crazy guy. It was like, because it's a very risky sampling. You're basically leaning into this carcass, which is full of maggots, and it's all bushes, and the tiger could be anywhere.
Yeah.
So there's always anywhere, even in Ranthambore, for example, when we sample, we are looking at the ground. We are always looking at the ground. We are not looking around. So we always have people who are also watching, just to make sure that it is dangerous in some sense. So basically, we didn't have any bad experiences, luckily. And I don't, generally, I don't know that much about tiger behavior, but they may not just attack you like that. They'll probably watch you for some time first. But yeah, so that is definitely something scary. And I think, it was a little bored, but I think once you get a hang of it, you can figure out how to do it without spending too much time at the kill.
Ma'am, I have no questions. It was very interactive session with you.
Yeah.
The first thing I would like to question is that how this molecular ecologist helpful to answer the solutions of human-wildlife conflict?
So that's what I was saying. So first of all, I think that it might be, so as of now, maybe Mr. Kale would be much better to answer this, but I think that, livestock depredation, it's difficult to avoid unless the livelihoods of people around protected areas do not include livestock. It's because the livestock is easy prey in that sense, right? So if there are individuals, there will be livestock depredation. But I think killing of humans is something really bad, and we should try to, address human kill as far as possible. So it would be really nice. See, getting to why it's happening, that is a very difficult question. I don't know if there's an answer to that.
How the molecular ecologist
That's
would
what I'm saying. Yeah. So basically, what I'm saying is, if there are individuals who are killing more, say, humans or something like that, those are potentially problem individuals. We can basically use these kill site sampling to identify who they are. And then they can be effectively removed from the population, taken into captivity or something like that. But there's no guarantee it won't happen again. It's a ongoing management intervention.
So there is any such markers like man-eaters, or we could identify?
No. So from our study so far, what we have found is there are no problem individuals. Many individuals are involved it's an ecological response. That is, there's abundant cattle, right? The densities of cattle are very high, and cattle are easier prey. From a perspective of a tiger, that would be an easier prey, and so it's an ecological response. It's not that there are specific individuals who are only killing cattle. Now, in the future, if there are families of tigers which grow up only eating cattle, it might become a social phenomenon. That is, cubs growing up only hunting cattle with their mother may not be able to go and hunt something which is harder to hunt. Something like this has kind of happened in Gujarat with the lions. There's a lot of carcass dumping, and as a result, a lot of the lions are just feeding on dead carcasses. They are not even hunting, right? So this is something which we think that we don't affect the wildlife, but even our practices are really changing their behavior. And so we don't know what will happen.
Okay.
Thank you.
If there are no more questions, I would like to thank Madam for a very engaging, interactive session. And I think as an ecologist, many of them sitting in the audience can relate to her love for the land and feel a very personal touch to the entire talk. And that really very well connects to the audience. And the experiences, many kind of experience where she shared in the very beginning of her talk are what many of the women scientists in STEM also face. So I think that is very well connected. Many of the women scientists and youngsters in the group, the non-invasive approach that she mentioned is kind of a breakthrough in wildlife. So I think that's very interesting to know, especially for scientists who are from environmental background in this audience largely. And this very interesting study of shoricolas, I think I recently heard V.V. Robbins. So this sky island is a very new terminology, and many of us might be interested in. This storytelling approach, Madam, has made it very exciting, interesting, and at the same time, a great learning and listening to all of us. 80% of genetic diversity in India and its future evolution potential is something very insightful to this audience. And your multiple contributions to different areas, human-wildlife conflict, addressing the One Health contest. I think this was a real package of knowledge in just an hour of session with so many questions that you have addressed. With this, I thank you, Madam, and I would request our director to present you a token of gratitude for this session. With this, we are towards the end of this very important session, and I'm sure a very exciting one that you all have enjoyed. I invite Mr. Prakash Kumbhare. Please give a formal vote of thanks.
I will not speak much, that I will ensure. You may be saying this paper, but I am not referring to that. That is answer. So, respected dignitaries on the dais and my colleagues and friends. Actually, what Madam's lecture was very encouraging, inspiring, and very informative science. So, what I suppose that we are doing ecosystem studies and natural resource problems, natural resource conservation. So, when she was explaining all those things for tiger, so I think DNA, Madam, can do much wonders, and it has done in the past also, and it is doing in the present also. What we have studied is from DNA only. So, but the important part was that I trust to my heart, it was without hurting an animal, means without hurting an animal or without touching. So they are collecting all samples, hairs and all. They are watching movement and all, where they are staying, where they are taking rest or something. And after that, they are going there and they are taking samples, and all the studies have been done with that only. That is the base. So that is because that systematic way they are doing, and they are this conservation genetics. They are so it is very important. I think from NEERI, from her lecture, what I take away is that we can also do such kind of things for our ecosystem conservation or any dealing with wild air, water, or something. So DNA is very important, and DNA is everywhere because microorganisms are everywhere. So that kind of thing, it is very important. And so thank you, Madam, for coming here and your wonderful lecture, because I enjoyed, we enjoyed. That is what you started for constitution to science, inclusivity, everything you have covered. I think nothing left. Okay. Sir, as usual, I have written sir in welcome address. So he told that from Brahman science, what is environment, agriculture, everything, medical science. So we are now enjoying that fruit which Dr. C.V. Raman had given earlier. So with this, I thank all my colleagues, all officials who were associated directly or indirectly, and everyone. I cannot tell name, and Madam particularly speaking. I will not take your name, Madam. Madam, I have told you. Madam means all Madam. Sirs means all sirs, because I cannot be very particular about any name. So thank you very much. And as usual, when I sit here, so my mind gets ignited, and 2 lines I have written in Hindi. I will end with that.
So madam has inspired me. So that lecture. So it is in Hindi, madam. Try to understand. I think you know Hindi, yes. महिला वैज्ञानिकों के साहस से, because madam has adventure. It is not because somebody has asked this question, how do you go and है ना? So जंगल में जाना और सैंपल लेना, शेर के मुंह में जाना, it is not very easy, है ना? तो मैडम ने भी बोला यहाँ पे कि मैं तो नीचे देखती हूँ, इधर उधर आई डोंट सी अराउंड, है ना? तो महिला वैज्ञानिकों के साहस से विज्ञान के राग, भारत बने विकसित उन्नति के साथ. थैंक यू वेरी मच. I forgot to stand for national anthem and with this will continue.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
थैंक यू. आप अपने सीट पे बैठ जाइए, पहले डिग्नेटरीज को जाने देंगे, उसके बाद फिर और रिफ्रेशमेंट लेके निकलेंगे सब.